Immunology Insights, Basics and Beyond. Chapter 7, Dendritic Cells. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our last chapter, we explored the roles of monocytes and macrophages, the frontline warriors of the immune system. Today, we are turning our attention to another crucial player in the immune defense, the dendritic cell, or DC. Known as the sentinels of the immune system, dendritic cells serve as the bridge between innate and adaptive immunity. They are the key messengers that alert the immune system to the presence of invaders, orchestrating a targeted and effective immune response. Understanding dendritic cells is essential because they are the primary antigen-presenting cells, or APCs, of the immune system, responsible for processing and presenting antigens to T cells. This function makes them central to initiating and regulating the immune response, but also makes them a therapeutic target. So let's talk science. Let's start with, what are dendritic cells? Well, dendritic cells are a type of white blood cell that originates primarily from the myeloid lineage, similar to monocytes and macrophages. They develop from hematopoietic stem cells in the bone marrow and then circulate in the bloodstream in an immature state. Although distinct from monocytes, certain dendritic cells can share precursors with monocytes and may differentiate in response to specific signaling factors. This background gives dendritic cells the ability to respond and process pathogens indiscriminately, akin to other innate immune cells. The name dendritic comes from the Greek word dendron, meaning tree, which reflects the cell's branched tree-like structure. These extensions, or dendrites, increase the cell's surface area, allowing them to capture and present antigens more effectively, as well as to interact with multiple cells. Now let's come to the role of dendritic cells in immune surveillance. Dendritic cells are constantly patrolling the body, searching for pathogens such as bacteria, viruses, and fungi. When they encounter a pathogen, they engulf it through a process called phagocytosis. Inside the dendritic cell, the pathogen is broken down into smaller fragments known as antigens. These antigens are then loaded onto major histocompatibility complex, or MHC, molecules and presented on the surface of the dendritic cell. Another important aspect is that dendritic cells function as a bridge between innate and adaptive immunity. Dendritic cells occupy a unique position in the immune system, linking innate and adaptive immunity. In their immature state, they act much like innate immune cells, indiscriminately phagocytosing pathogens and debris. However, once they capture an antigen, dendritic cells mature and can then teach the adaptive immune system by presenting these processed antigens to T cells. This presentation on both MHC1 and MHC2 molecules activates T cells, initiating an adaptive immune response that is highly specific to the detected pathogen. This pivotal role makes dendritic cells indispensable for triggering a targeted, memory-forming immune response while also guiding its specificity and scope. An important distinction here is that dendritic cells can present antigens on both MHC class I and MHC class II molecules. MHC class I presents antigens to CD8 positive T cells, or killer T cells, which often respond to intracellular infections like viruses, while MHC class II presents to CD4 positive T cells, or T helper cells, which are central to coordinate broader immune responses. Let's now come to maturation and migration of dendritic cells. Once a dendritic cell has captured an antigen, it undergoes the process of maturation. During this maturation, the dendritic cell upregulates the expression of co-stimulatory molecules and MHC molecules, essential for effective T cell activation. These mature dendritic cells then migrate from the tissues where they captured the antigen to the nearest lymph node, a process guided by chemokines, which are signaling molecules that direct the movement of immune cells. The lymph nodes serve as meeting points where dendritic cells present antigens to T cells. This interaction forms the so-called immunological synapse, a structured interface where signals are exchanged between dendritic cells and T cells to fine-tune the immune response. Additionally, mature dendritic cells can also interact with B cells, providing signals that help activate and shape the antibody response, adding another layer to the immune defense. There are several subtypes of dendritic cells, each with specialized functions. First, we have the conventional dendritic cells, or CDCs. These are the most common type and are highly efficient at capturing antigens and presenting them to T cells. They are further classified into CDC1 and CDC2 subtypes, each of which is specialized in presenting antigens to different types of T cells. Second, we have the plasmacytoid dendritic cells, or PDCs. These cells are primarily involved in the antiviral immune response. They produce large amounts of interferon alpha, a key antiviral cytokine in response to viral infections. Third, we have the Langerhans cells, a specialized type of dendritic cell found in the skin. Langerhans cells are particularly important for the immune response to skin infections and for maintaining tolerance to harmless environmental antigens. Each of these dendritic cell subtypes plays a unique role in ensuring that the immune system responds appropriately to different types of pathogens. Next, we have the role of dendritic cells in immune tolerance. While dendritic cells are critical for initiating the immune responses, they also play a key role in maintaining the immune tolerance, preventing the immune system from attacking the body's own tissues. In the absence of infection, dendritic cells present self-antigen to T cells in a way that does not trigger an immune response. Instead, this interaction promotes the development of regulatory T cells, which help to suppress potential autoimmune reactions. This balance between activating an immune response to pathogens and maintaining tolerance to self-antigens is crucial for preventing autoimmune diseases, where the immune system mistakenly attacks the body's own tissues. There are also diseases and disorders linked to dendritic cells. Disruptions in dendritic cell function have been associated with a variety of diseases. For example, an overactive or improperly functioning dendritic cell can contribute to autoimmune diseases, such as lupus or rheumatoid arthritis, where the self-antigens are mistakenly treated as threats. Dendritic cells are also implicated in certain cancers, where they may fail to properly activate T cells, allowing tumors to evade immune detection. All this makes dendritic cells an interesting target for therapeutic applications. Given their central role in immune activation, dendritic cells have become a focal point in the development of immunotherapies. In cancer immunotherapy, for example, dendritic cells can be engineered to present tumor antigens to T cells, enhancing the body's ability to recognize and destroy cancer cells. Dendritic cell vaccines are being explored as a treatment for various cancers, including melanoma and prostate cancer. These vaccines involve isolating dendritic cells from a patient, loading them with tumor antigens, and then reintroducing them into the patient's body to stimulate a robust anti-tumor immune response. In addition to cancer, dendritic cells are being studied in the context of infectious diseases, autoimmune disorders, and organ transplantation. By modulating dendritic cell function, it may be possible to enhance immune responses where they are needed, or to suppress them in conditions where the immune system is overactive. To conclude, dendritic cells are the unsung heroes of the immune system, serving as the crucial link between detecting a pathogen and mounting a targeted immune response. Their ability to process and present antigens makes them indispensable in the fight against infections, cancer, and even in maintaining immune tolerance to prevent autoimmune diseases. As we continue to explore the immune system, the importance of dendritic cells becomes even clearer. Their role in both activating and regulating the immune response underscores the complexity and precision of our body's defense mechanisms. In our next chapter, we will turn our focus on natural killer cells, another fascinating component of the immune system that plays a critical role in the early defense against viruses and tumors. Until then, remember that these dendritic cells are constantly at work protecting you by ensuring that your immune system is always prepared to respond to threats. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So see you next time and thank you for listening.